We have seen the process flow chart for initial preparation of the manuscript. Uh, Before that, in the preparation list, I have mentioned about data compilation and arrangement. This is a very important part. So whatever the data which you have generated has to be arranged in this following order, that is according to introduction, then materials and matter. So in introduction section you had to write about the problem to be discussed, as well as a review of literature. So whatever the references which are mentioned in literature, descriptions which are mentioned in literature about that particular investigation topic, that you have to write in an introduction section. Then whatever the methodologies which have been carried out should be written in materials and methods, then results and references. So you should keep this particular material ready in this particular order. Then the most important thing is to prepare tables and figures. You see, basically, this gives the idea about your work. That means, have you completed your, even if you have completed your objectives, but the amount of data that you have generated, so you can predict that from the number of tables and figures that can be generated through this particular data. So basically, why this part is important, preparation of tables and figures, you should take this task in the beginning, because this will give you idea about your work, whether the sufficient amount of work is done which is sufficient enough to write a project report or a paper. So approximately, according to my study, I think approximately 15 total, in all 15 figures and tables, including images or photographs. So this should be the minimum output. It can be anything, it can be more than that, but minimum output it should be 15, in all 15 figures, tables and images. This amount of work is, I think, sufficient enough for writing a completion of the project or writing a research paper. So this is how we go for the preparation of the first draft. And how do we prepare the first draft? We prepare the first draft according to the first step, that is arranging your material in the order of introduction, materials and methods, results and references. And then we go for searching for the journal. So when we select a particular journal, then according to the guidelines of that particular journal, you have to write down or you have to rearrange the data. So that will be called as a first draft of your manuscript. Searching for suitable journal, this is again a very critical thing, and it may take uh, several days in order to search for a suitable journal. So while searching for suitable journal, the question arises how to select a particular journal. So there are several quality measures which are mentioned in this slide. First, you should search for journal related to your subject area or the scope of your research. So according to the subject of your research which you have carried out, accordingly, for example, I have carried out my work in microbiology, so I will go in the search of microbiology journals. So according to your subject, so in microbiology also there are specializations, like agricultural microbiology is there, fermentation technology is there, genetics is there, plant biology is there, soil microbiology is there. So accordingly, there are specialized journals for all these different topics, so we can search for such journals. Second quality measure that you should look for, that whether that particular journal is listed in UGC care list, which is being prepared by SPPU university. Then whether the journal is indexed in or it is listed in Scopus. So now Scopus, this is a type of database which is prepared by Elsewhere. So it is Elsewhere's abstracts and citation database that gives several quality parameters. For example, H index, site score, scientific journal ratings, and SNIP. So based on this, so if the journal is listed in Scopus, of course it shows its reputation. Then the journal should be listed in Web of Science. This is again a database, WOC. Then the journal should be a peer reviewed journal. So I think we'll discuss more about peer reviewed journals in the next few slides. And the most important quality measure that decides the reputation of that particular journal in the scientific community is its impact factor. Now there are several ways of calculating impact factors. Someone else may be talking on that, but which impact factor has to be considered? So if it is calculated by means, or if it is mentioned in the Scopus, then that impact factor is considered as the authentic one. Remaining impact factors doesn't seems to have a significant value. Then you can go for, while selecting a journal, you can see what is who is the publisher of that journal. So these are the few reputed publishers. Publication in Elsewhere, Taylor and Francis Group, Hindvi Publishers, Springer, all these are the reputed publishers. So publication in these journals is again one of the quality measures. So for example, this is a journal of which is published by Elsewhere, that is Bioresource Technology. This is basically for this is a multi-disciplinary journal, we can say. Chemistry people, botany, zoology, mathematics, or something, microbiology. So whatever the subjects which have a little connection with biology, they can publish their work in Bioresource Technology, which is having a very high impact factor of 4.5. So while selecting a journal, for example, I have opened this for Bioresource Technology. So when I open this, this is something what is displayed. So this gives the description of the journal. Then what are the guidelines, impact factor, abstracting and indexing, and what is the who are there in the editorial board. So that is again a very important criteria to be considered. That is who are the contributors in editorial board. So you will find a description of description means this gives you the scope. That means what type of articles are published in this journal, and what are the subject areas which are considered for the publication in this particular journal. So in this you can see that this journal is involved in, or it is related to, the publication of data in the areas of biomass, biological waste treatment, then bioenergy, biotransformations, bioresource system analysis, and different types of technology. So this is how you come to know regarding the scope of this particular journal. So according to your subject area, you can choose the journal. This is just an example of that. Then this journal page will give you the details about abstracting and indexing. You can see here that this journal is abstracted in several databases. So for example, more than 30 we can see here. So that shows the reputation of this journal. So important ones are whether it is indexed in PubMed, Scopus, Elsewhere database, etc., etc. And this page also shows you the who are there in what is the constitution of this editorial board for this journal. Then there is a database called as Clavierate Analytics. that is this is involved in the analysis of journals and deciding the quality of the journals. So you can see here, you can get the list of important journals from this particular database. So you can see here there is a list of journals showing the title, then site score, and you can see this 4 column, that is journal impact factor. So that is what we have to consider, that shows the actual rating of this journal. You can see that the first journal, that is cancer journal for clinicians, it shows an impact factor of 224, which is a huge impact factor. So this is how you can see the name of your selected journal in the list, and you can see what is the impact factor. Then there are several Indian journals in which you can publish. So these journals are listed in Scopus. Of course, there is a separate website for Scopus. So if you just type this keyword of Scopus in Google, then it will take you to the website of this Scopus, where you can search for journals which are indexed in Scopus along with their impact factor. This list I have obtained from UGC Care. So that is a link that you can get from SPPU website. So you have to just type UGC Care list, then you have to sign up, then log in, and then you can obtain this list from which is prepared by SPPU for the UGC Care list. So you can see the list of journal and publisher along with their ISSN and ISBN number. After the selection of journals, now let us move on to the full-length research paper, writing a full-length research paper. So I'll be showing you or explaining you this with the help of a sample paper. So in general, this original manuscript, which is a descriptive one, as we have seen earlier, it consists of an abstract, then keywords, introduction, materials and methods, results and discussion, acknowledgement, and references. So this is in general standard format for any full-length research paper. The E1, that is results and discussion. Some of the journals, they prefer that results and discussions to be written in combination, in a combined form, or some journals prefer the separate sections of results and discussion. So there will be a separate result section and discussion section. So before we go for the preparation of manuscript, you need to read very carefully the instructions regarding the preparation of the manuscript. So in instruction part, you will find what should be the structure of that manuscript, means the order of the titles, the limitation of words. So this differs from journal to journal. So total number of words, then what should be the style of the title, how to write the author's names and its affiliations. All these guidelines are given. Then regarding the corresponding author, you have to mention its email, then how to write the abstract, what are the word limitations, what should be the font size for writing a title, what should be the font size, style and spacing for writing the author's names, affiliations, as well as the introduction section and several other sections. Then it will of course mention that the use of tab stops for indents and not to use a space bar. So all these minor details will be mentioned in this section of instructions to authors. Then regarding the use of equation editor or math type equation. So in general, manuscripts with mathematical content that can be submitted in latex format. So then it mentions about what should be the size of the image, resolution, and labeling, figures, instructions regarding figures, table captions, so etc. So all these instructions you need to follow. And finally, what should be the style of references? References means literature that you have referred, that should be mentioned in text, and end references also, because if you are using the reference or the data, image or information or the sentences of somewhere else, means if the work is already published and you are using that those particular set of words, images, for the sake of references, then you need to uh, mention about that. So that is just to avoid the plagiarism type of problems in your manuscript. So we should acknowledge. So that is a way of acknowledgement. Then while writing a title, there is a standard format for that. It should be short, it should be non-descriptive, it should be informative enough to give the idea of that particular research which has been carried out by you. It should be concise and of course meaningful. It should reflect your work. So means reading a title, it should give idea to the reader what work exactly has been carried out by you and what is its significance. It should also reflect the novelty or newness of your work. So in short, we can say that the title should be such that it should give idea about all these mentioned parameters. Just an example, you see this title, it is long enough, I think. So in general, the title can be from 6 to 20 words. So I think this title is approximately less than 20, so it is fine. So let us see what is this title. It is Nano Silver Incorporated Biomimetic Polydopamine Coatings on a Thermoplastic Polyurethane Porous Nanocomposite as an efficient antibacterial wound dressing. So this title gives you the idea regarding what work, significant work has been carried out. That means this title something gives us idea that uh, this research is involved in the preparation of antibacterial wound dressing, and the materials which are used for the preparation of this antibacterial wound dressings are nano silver incorporated biomimic polydopamine coating, which is one of the unique words. So this is how the title should be. There are several examples. I can tell you that this is the second example. Biology students may understand this. In general, biological control of oily spot disease of pomegranate by antibacterial lactic acid bacteria, one of the simple titles that tells us that this work is related to the control of a disease of pomegranate by lactic acid bacteria. Then the title, it can be simple as ecology and biodiversity of cyanobacteria from saline soil. So this is an example of the simple titles. Another example is ecology and biodiversity of nitrogen fixing and phosphate solubilizing bacteria from saline soil, extraction of polychemicals. So last three titles are not that much significant, I think, according to me. It should not be very lengthy, of course. Then numerical approximation of non-isothermal two-dimensional general rate model of reactive liquid chromatography. So this title gives an idea regarding the work that liquid chromatography has been employed for this particular two-dimensional preparation of two-dimensional general rate model. Then this is how we can go for the making a title. There are several examples you can see on internet, on Google Scholar, ResearchGate. So as I said, approximately the word limit is 7 to 20 words, and you need to avoid the words like a study on, or a study of, or an investigation on, or an investigation of, studies on. These are very common words, and these are this makes your research very general. So your research title has to be very specific. So you try to avoid all these titles. So after writing title, the next thing you have to write in a research paper is authors names. Authors names, what are authors? Authors are the contributors to that particular research. So whoever has taken efforts in experimentation, investigation, examination, or survey. So all these authors, all these researchers, workers will be named as authors. So there can be a very short list. It can be a single author, two author, three author. If the work is very big, there can be 20 authors, 25 authors. So it is dependent on the nature of the work, or the in how much. So this is related to how big is your research work, how many institutes are involved, how many different subject experts are involved. So likewise, so this is just an example. You can see this is a paper. So all these authors are from China, Menglong, Liu. So in general, what we write is the surname. We write the surname first and the name first. So for example, if my name is Milin Gajviye, so I will have to write Gajviye Milin. Or sometimes journal will ask you to write Gajviye MH or Gajviye M, followed by that the next author. You can see that there is a superscript written on the head of each author. So for example, the first author is Liu, then you will find there is a superscript written 1. So likewise, 1, second author is 1, third author is 1. So what is all this? So all these belong to the first institution, and you can see there is 3, so they belong to third institution. So this indicates the name of the institute. Then there are so many authors, so what is their contribution? So you can separately mention that first 3 authors, or specifically you can name that author, that this particular author carried out this particular work. So this is how you can categorize the contributions of the authors, which can be of course mentioned in the manuscript. In general, first author has the highest contribution, that is 70% of the contribution. And you can see this last author, that is Jun Wu. You can see there is an asterisk, there is a mark on which is written in the superscript. What is that indication? That indicates the author is a corresponding author. So this corresponding author is responsible for the submission and the total correspondence which he is going to make with the related journal. So corresponding author and first author are significant, and their contribution is considered as 70%, 70. Rest of the authors are called as co-authors. So all these co-authors are going to get 30% of contribution unless and until it is mentioned separately in the manuscript. So if someone wants to deny 30% contribution and he or she is a contributor, equal contributor like first author, then that should be mentioned in the manuscript, that first 4 authors have equal contribution in this manuscript likewise. For example, sometimes it may happen that a particular researcher has done some significant work, or it has some contribution in that particular research, but somehow by some means his or her name is neglected from the list of authors while submitting a manuscript. So what are these authors? These authors which do have actual contributed in this particular research, but their contribution is denied or neglected due to several reasons. The reasons may be controversial reasons. So such type of authors are ghost authors, and that is an illegal thing. It is not ethical. So I think ethical guidelines, of course someone else is going to talk on that, so I'm not going to spend much time on that. So this is something related to the ethics. So this should not happen. The ghost author should not be there. You need to mention the name of all the authors who have contributed in this particular in your research. Then there is another category of authors that should be avoided, that there are some authors whose names are included in the list of authors, in the list of contributors, but at actual these authors haven't at all contributed. There is no contribution of these authors, and still they are enlisted. Why they are enlisted? Because of the influence. Say for example, you cannot avoid the inclusion of name of your head of the department, head of your division, your principal, likewise, so because of their pressure, you are forced to add their names. So again, that is an illegal thing. It is not ethical. Such authors are called as gift and guest authors. So these two authors doesn't have actual contribution, but they may be added, but as I said, it is not ethical, so it should be avoided. So that is regarding the authorship. The next thing you have to write on the title page is affiliation. So as I mentioned in the earlier slide, there was a superscript 1, 2 and 3. So that is the affiliation of the authors to which they belong to. So 1, 2, 3. Likewise, you have to mention the name of the institute, the place, the pin code, and the name of the place, state, as well as the country. You see the list of authors. The last author showed in superscript and asterisk, so that mentions about the corresponding author. So you have to mention the email ID of corresponding author. So as I mentioned, corresponding author will be responsible for all the communications and correspondence with the journal. Then after affiliation, what we write next in the paper is an abstract. What is an abstract? So abstract has to be written in limited words. So this is according to the journal guideline we have to write. Some journals, they have they give a limitation of words of 150, 200, 250 and approximately 300 words. So what is an abstract? It is a concise description of your findings or investigation. So whatever the work which you have carried out, or whatever the work that you are describing in the latter parts of the paper, so the concise paragraph you have to write in the form of an abstract. So it is a brief summary, we can say, that should describe the major findings. So that should the abstract should consist of background information, the problem which is being investigated, then what are the major findings of investigation. Mostly it should be written in numerical form, or you need to describe the data here. So it can be written in the form of aims, methods and results, conclusions, significance and impact of this. So all these points should be covered in limited amount of words, that is called as an abstract. So a reader will get idea exactly what work has been carried out. This is just an example of this is the journal of nanotechnology. So according this abstract is prepared according to the instructions of that journal. So that journal prefers the writing of abstract in this manner: background, results, conclusion, and later on at the end keyword. So keyword, it is not the part of abstract. Keywords are written separately. So you can see, you can get idea about how to write abstract from this. So in the first paragraph, background, the introductory part and the problem that was undertaken, it is written. Then the results are written in numerical form, and you can see that there are several words. You can see SEM images. So that just gives the idea that what kind of techniques, technologies have been used, like scanning electron microscopy has been used in this investigation. So in the result section, they have written the results, and in order to investigate, in order to obtain those results or findings, what methods that they have used, it is also written. And finally, the inference, or what is the output or the final conclusion, it can be written in the abstract. There is a second type of abstract. Some of the journals that they may prefer, like bioresearch technology, they will ask you for submission of graphical abstract. So one way of writing abstract is in text, as the previous slide shows. The another way is a graphical abstract. That means you can show the abstract in the form of a diagram. This diagram shows what is the title, what are the objectives, what are the methodologies which are used, what is your result, and what is the novelty. So in this graphical abstract, you can see that there are several diagrams which are shown. So these diagrams, few diagrams, like you can see the tube, flask, and the column chromatography. So that shows the techniques, unique techniques that have been used. You can see here at the bottom there is a plate showing a white part. So that white part is fungus, and the clear part is the fungus. The growth of the fungus is being inhibited. So that is the result. You can see several formulas here. So that is the part of result. GCMS analysis, that is a part of technique which is used in this analysis. And at the end, it is written that it is the first report of novel antifungal compound. So this is the newness of this manuscript, or this is the novelty, or this is the conclusion of this manuscript. So this is how you can write the graphical abstract as well. Then after writing abstract, you need to write keywords. So in general, the keywords are 5 to 6. So keywords, listing of keywords is very significant, because keywords are responsible for, as the name indicates, key means significant, important words. These words are going to be useful for the readers in order to search information about your research paper. So a list of approximately 5 to 8 keywords should be added. So these keywords are basically, if you put this keyword in Google, so immediately your paper should be displayed, should be like that. So these keywords are important to unlock the information so that it will be available for the referencing by the others. So basically the idea is that these keywords should not be, so if you are interested in finding out your article, so what we can do is directly we can copy paste this title in Google, So another way is you can copy paste keywords in Google. So in general, the keyword should not be repeated. So whatever the words which are there in title should not be repeated as keywords. Are you understanding? So it should be a complete set in order to uh, be made available for the referencing by the readers. Then the next section what we write after keywords is an introduction section. So introduction section, it gives the background information. It is a brief description of the problem that is to be investigated or addressed, information to introduce a particular subject, along with or taking support of several references, citations of relevant literature or references. So if you are writing some sentences, information, so to support that sentence, what we write is the reference. So in the end, I'll tell you how to write the or cite the references. So the preliminary background information should be written in introduction section. Then it can be explained, the problem can be explained with the help of figures or diagrams. So use of diagram and figures is allowed in introduction section. This introduction section should incorporate the hypothesis. So what is your hypothesis? What problem on which you are investigating, that should be mentioned. It should clarify the aims and objectives and the purpose of your work. So all these things should be included in an introduction section. After writing introduction section, the next part is writing materials and methods. So whatever the materials which are used, for example, it can be a chemical, it can be a plant material, animals may be used in this study, different equipments may be used, microorganisms, database, software, equations, etc. So whatever these things are there, they are called as materials. So they should be mentioned, properly mentioned, described in materials and methods sections. Of course, this part will describe the methodology or the techniques or the protocols or the procedures that have been used. So procedures and protocols should be written such that anyone, means the reader, should be able to repeat that particular experiment in his or her laboratory. So sufficient details should be provided so that the experiments can be repeated. Then if some references are available, if the technique has been discussed earlier, you can put acknowledge the work of that by putting the reference. In materials and methods sections, there can be the use of headings. So it can be headings or subheadings. So we call them as the labels. So we'll see that in next few slides. After writing materials and methods, the next part we write is a results section, results and discussion section. Now, as I mentioned earlier, it can be a combined section or it can be a separate for results and discussion. So what we write in results and discussion is well prepared tables and figures. The figures may be images, photographs, etc., etc. So this must be a cardinal feature of the results section because they convey the major observations to readers who scan a paper. That means your results should include tables, figures, because what readers are going to see first is not what is written in the text or what is described in the paper. So basically they are going to look at your figure, table. So figure talks or the table talks more than what is written in the description of the results. Discussion section, it can be separate or in combination with results. As I said, what is a discussion? This is one of the very critical things sometimes it may be difficult. So discussion is comparison of your findings, whatever the results that you obtain during your investigation, examination, survey. So whatever your findings are there, so you have to compare your findings with the others findings. So already literature is published on this related work, so what are their results and what you got during your investigation. So it may be controversial, it may be the findings may be in line or in agreement with the other publishers or other workers, or it may be opposite to that. So all these comparisons you have to write in descriptive manner in a discussion section. And while discussing, if you are taking reference or reference of the other's work, then you need to cite that reference in your discussion section. So cite the relevant literature and references. Then next part we write is about the acknowledgement. So acknowledgement is to for mentioning the contributors. So there may be many indirect people who have indirectly helped you in performing this particular investigation or survey. So we cannot give we cannot mention them in the list of authors. So how to acknowledge them is you can mention these contributors in final acknowledgement section. So these are the contributors who do not qualify as authors, so you are not listing them as authors. So they should be acknowledged, and their particular contribution should be described in the acknowledgement section. So what it includes all the sources of funding for the work reported for all the authors. So whatever the help, financial help, funding that you received by that is received by any of the author, that should be acknowledged. Both the research funder and grant number. So this is related to if you get a project from funding agencies, Indian funding agency or foreign funding agency, then you will get a grant number. So that should be mentioned that you receive fund for this particular research work. For you can mention if you have received any financial assistance. Like in college we have student research project scheme, so college provides funds. So if you are going for publication in relation to this work, so you should mention the name of the college that you received funds for in order to carry out this particular research from the college. Then you may have received any technical assistance, like some of the techniques or some of the instruments which are not available in college in your institution, so you hire them free of cost from some other institution. So you, for example, if you want some instrument like FTR, if it is not available in our institution, so you go to the next institution, use that facility. So you can mention that you have used that facility, and you acknowledge, you can say thanks to the institution for making that particular facility available to you. So likewise, scientific help, technical help, you can mention in the acknowledgement section. And of course the last section will be the references, that will be called as the end references. So the next few slides I'll be explaining you what are the styles and how to write the referencing section. This is just an example of a sample paper of original research paper. You can see this is my paper published in Proceedings of National Academy of Sciences, India, Bioefficiency of Antifungal Lactic Acid Bacterial Isolates of. So this is a Springer publication. There are two authors, myself and my guide. You can see that in the first section there is an abstract of approximately 250 words. You can see that there are numerical figures 224, 360. Can you see that? 81.6. This is called as a numerical data. So this is very significant that you need to mention the numerical data. Then you can see the name of the organisms. So this is about the findings. You can see several values in there. And finally, can you see in the next column in bracket it is written P 0.05. So here you can see that I have made use of a statistical tool, a statistical test for the significant difference. So this adds or this increases the potential of your manuscript, of course, the use of statistical tools. And finally it ends with thus pomegranate is the source of. So that is the conclusion part of that. Then it is followed by the keywords. Now here I told you earlier that the words should not be repeated, but unfortunately, being a preliminary researcher that time, I repeated. So that mistake you should not make. I have made that mistake. Then introduction section, you can see that introduction to this particular topic is written. Can you see in brackets, square brackets, the numbers are written 1, 2, 3, 5. What are all these? This is a way of writing the references. I'm gonna tell you regarding how to write or how to cite the references later on. So this is one way of citing the references. So end references I have written at the end of this manuscript. They are written in a list in a numbering system 1, 2, 3, 4, and that number I have cited here. That means I have used that reference in order to write this particular paragraph or in order to write that particular sentence. So this is how we have to write. So as I mentioned earlier, that is the abstract is a short description. Then introduction is the background information you have to write. You can see this is another sample copy of introduction section. You can see in blue in square brackets references are mentioned. You can see the second column. The red arrows. Can you see the red arrows? So here there is a mention of abbreviations. Okay, thermoplastic polyurethane. So this word, I think the author want to use it more frequently. So this word is going to come repeatedly in the manuscript. So if you going to use this complete word repeatedly, then what will happen? What is going to happen? The word limit of manuscript is going to be increased. So instead of repeating that word, we can make use of its abbreviated form, that is TPU. Likewise, you can see several short forms. What you can do here is you have to use the full word in the beginning and give a short form of that so that you can use it repeatedly. So you can see here PD, DA, these are the abbreviations that will be used in the manuscript to describe the full forms. Okay, likewise. Sometimes you need not to mention the full form of few standard words. Standard word means here you can see SEM. So it is a standard technique which is known to the scientific community. So you need not to put long form of SEM. So SEM is scanning electron microscopy. So these are the standard word which you need not to describe. Then if you see this paragraph that starts with in this study. So this is the last paragraph of introduction section. So as I mentioned earlier, this last paragraph should mention about your objectives, what work you have carried out. So you can see that with the help of diagram also you can justify or you can describe your background information or introduction section. Then Materials and methods. So as I mentioned, materials and methods, it may have several headings. You can see that headings are written under materials and methods sections in bold. So materials and methods section should describe the materials, and while writing materials, you need to mention about the, for example, if you are using a chemical, so you need to write what is the company of that particular chemical. So here it is written Sigma Aldrich, from and from which country it is obtained or you have used. So you need to write the country, place, and the make, that is Sigma. Likewise, these are the heading labels. As I mentioned, these heading labels may be first level, second level, third level. So if there are 3 heading labels, then first level heading can be written in bold, second level heading can be written in bold italics, and third level heading it can be written in only italics. So this is just to differentiate the different headings. Then again, regarding the use of units, this is again very important to use the units. This should be written according to the guidelines of the journal. So most of the journals prefer SI, writing SI units. So it can be written in short forms, like for grams it is G, for milliliters it is ml, it can be m, small m, capital liter. So according to the guidelines of the journal, you need to write. This is very important because this is a part of scientific writing. This is the part of scientific writing which is very important. So you may use several symbols for the units. Likewise, it is mentioned for liters, it may be small l or capital L. So you will get idea about this in the instructions to authors. Then the next part is results and discussion. So results and discussion may or may not have the headings. So under here there is a heading of morphology and character. So that is a heading. So you can describe results and discussion in combinations with different headings with the help of diagrams. So diagrams are really very important. So whatever you have written in this results section, whatever you have written in the results section, so that description should be reflected in the figures. So these are the diagrams that show scanning electron micrograph. These are the images, photographs. You can see the graphs, you can see the ups and downs, peaks. So all this description is there, you will find in the results section. Next, importance of figures are really very important. So I found this on Google, that a picture worth 1000 words. So as I mentioned earlier, instead of reader is going to look at your table and figure first, instead of reading the big description in words. So this picture is very significant one. So it should give all the information which is written in the text. Figures are of different types. Now you can see in this figure, this is a single figure, but there are several subsections which are mentioned as A, B, C, D, E. So these are several subsections. You can mention these subsections in this is called as, in blue, what is written, the blue box, it is called as a figure caption. So writing a figure caption is again a very critical thing. It should give a brief idea about whatever is shown in the figure. So writing figure caption is again a part of skill. So figure caption, it describes what is A part, what is B part, and so on, what are E parts. Resolution of figure, it should be clean. In general, it should be between 300 to 1200 dpi. figures are to be supplied electronically. That means you need to upload a separate file of figure in the form of TIFF, JPG or PNG, that we'll see later on. Then there can be the figures like a bar diagram. This is really very important. Many of you may be going to prepare the bar diagrams. You can see that in order to increase the value of your bar diagram, what are the different tools? So the very common tool is a statistical tool that you have to use. You can see in this first bar diagram, the x-axis is post inoculation days. That means these are the days, and on the top of these columns you will find a letter. So what is that letter? So that letter tells you about the level of significance. That means that gives the reader idea that you have used some statistical tool in order to analyze this particular data. So at the same time, can you see this small stick on the head of this column? So that is called as the error bar. So error bar means this shows that you have replicated, you have used several replication. That means you haven't performed this experiment only at once, you have repeated this experiment more than 3 times. So that is also very important. Can you see this symbol that is marked on 10th day, and on 10th day you have 1 2 3 4 5 columns. Can you see that there are 5 columns? 1 2 3 4 5. That means there are 5 groups which are analyzed on 10th day. So these groups, or these are the different trials. These are different trials, and you can see, and these trials have been analyzed by using a statistical test called as say for example statistical test for significance. So in this study I have used Tuckey's HSD for statistical significance. That means these, whether there is a significant difference between these groups or not, so that can be analyzed by using this statistical Tuckey's test. So I have used that, and what you find is in first 3 columns at the top of that you can see AAA. So if the letters are same, what does it mean? It means that these groups doesn't have any significant difference. Fourth column is B. This means that there is a statistical significance, or there is a difference between first 3 groups and fourth group. So fourth group is different. And the next column, the last one, it is C. So this means that different letters means there is a significant difference. Same letters, there is no significant difference. So I am telling you this because this is going to enrich, or this is going to make your figure rich, scientifically rich. Therefore use of statistical techniques is very important. The figure should be self-explanatory. That means the reader should not go back to the text, whatever is written in text. So you should he or she should understand just looking at the figure. This is the another format of figures which is called as a line figure. You can see here smooth lines. There can be use of straight lines. You can see that there are 2 y-axis and 1 x-axis. The reason behind that is we have several parameters to be displayed on a single figure. You can see that on x-axis, on y1 axis, there are 3 parameters: pH, antifungal activity, and inhibition zone diameter. So there are several lines, so reader may get confused. So in order to make it easier, you can see that there are several legends like dots, triangles, hollow triangles, and filled triangles. You can see that there are circles, filled circles. Filled circles That means it is a symbol of that describes the pH. So you can see here, and that is also mentioned in the figure caption. So this makes easier. So one thing is missing in this figure. Can you guess? So that thing is the bar, error bar. So if you put error bars in this diagram, that will definitely going to increase the value of this figure. Then there are tables. Preparation of tables is again a very crucial thing. The arrow indicates the information which is written at the bottom. It is written in superscript and it is called as a footnote. Footnotes should be indicated by superscript or lowercase letters, and they can be symbolized by asterisk, hashtag, which are mentioned likewise. So you can put that in front of any word, and what is the meaning of that you can put as a footnote like this likewise. So it is supplied at the end of the table.